Hello, myself Vaishnavi Khamkar from PES Modern College of Pharmacy, Nigdi. My topic is IR Spectroscopy, subject Instrumental Method Analysis, guided by Dr. Aishwarya Balap ma'am. And my presentation colleagues are Vaishnavi Katkhede, Janhavi Katkhede, Rutuja Khandare, and Shravani Khedakar. Firstly, we see the principle of IR spectroscopy. Molecules are made up of atoms linked by bond. The movement of atoms and the chemical bonds like a spring and ball, stretch, compress, equilibrium bond length. These characteristic vibrations are known as a natural vibrations. The energy of molecular vibration is quantized. Let's see the introduction to IR spectroscopy. IR spectroscopy is an analytical technique used to study molecular vibrations. It measures the absorption of infrared radiation by sample as a function of wavelength or wave number, primarily used for qualitative and semi-quantitative analysis of organic and inorganic compounds. Next, criteria for a compound to absorb IR radiation. Change in dipole moment. First, a molecule can only absorb IR radiation when its absorption causes a change in its electric dipole. Second, a polar bond is usually IR active. Third, a non-polar bond in a symmetrical molecule will absorb weakly or not at all. When EMR is applied, then it causes the vibration between the atoms of molecule. When apply inferior frequency equal to natural frequency of vibration, then absorption of IR radiation takes place and peak is observed. Stretching and bending vibrations. Stretching vibrations involve a continuous change in the interatomic distance along the axis of the bond between two atoms. It requires more energy to appear at shorter wavelength, while bending vibrations are characterized by a change in the angle between two bonds. It requires less energy to appear at longer wavelength. Types of molecular vibrations. There are two types: non fundamental vibrations and fundamental vibrations. In fundamental vibrations, stretching vibrations and bending vibrations. And in non fundamental vibrations include overtones, Fermi resonance, combination tones. Let's see the bending vibrations. There are two types: in plane bending and out of plane bending. In plane bending includes scissoring and rocking. Out plane bending includes wagging and twisting. Stretching vibrations. There are two stretching vibrations: asymmetric and symmetric. Asymmetric is a interatomic distance between two atoms is opposite, and in symmetric, interatomic distance between two atoms is increases or decreases. Bending vibrations. These are characterized by continuously changing bond angles and axes with common atom. These are of various types, as positive indicates movement out of the plane above and negative indicates movement back of the plane. Scissoring vibrations occurs when two atoms move back and forth towards each other, while rocking vibrations occurs due to oscillation of atoms and further out of equilibrium plane. Hooke's law for frequency calculation. The probable frequency of absorption can be calculated by Hooke's law. Theta is equal to 1 divided by 2 pi c under root f divided by m underscore x m underscore y divided by m underscore x plus m underscore y, where theta is the vibrational frequency and c is the velocity of light, f is a force constant of bond, m underscore x and m underscore y is a mass of atom x and atom y respectively. However, it has been observed that the calculated value of frequency of absorption is not exactly equal to the experimental value. There are many factors which are responsible for shift in vibrational frequencies are as follow. Bending vibrations. Wagging vibration results when the unit oscillates in equilibrium plane formed by atoms. Twisting vibrations occurs when the structure unit rotates around the bond which join to the molecule. Here is a graph which explains the IR spectroscopy. Let's see the application of IR spectroscopy. Identification of substances. To compare spectrums, no two samples will have identical IR spectrum criteria. Sample and reference must be tested in the identical conditions like physical state, temperature, solvent, etc. The fingerprint region. Small differences in structure and constitution of molecule results in significant changes in the peak in this region. Hence, this region helps in identify an unknown compound. Studying progress of reactions, observing rate of disappearance of characteristics absorption bands in reactants, rate of increasing absorption bands in the product of a particular product, measure the degree of polymerization in chemical compounds, determination of molecular structure used along with the other spectroscopic techniques. Identification is done based on the position of absorption bands in the spectrum. Absence of bands of particular group indicates absence of that group in the compound. Here is a wave number and absorbance graph of unknown and benzene sample. Detection of impurities determined by the comparing sample spectrum with the spectrum of pure reference compound. Example, ketone impurities in alcohol. Detection is favored when impurity possesses strong band in IR region where the main substances do not possess band. Example is impurities in the beeswax with the petroleum wax. Monitoring the structural plasticity of plant cell walls. Here is a carbohydrates and their wavelengths. Next is a protein quantitation. IR spectroscopy is one of the most well established technique for the analysis of protein structure. In protein, amino acids are covalently linked via amide bonds. It absorbs in multiple regions of the mid-IR spectrum. By measuring amide bonds in the protein chain, we can accurately quantify an intrinsic components of every protein. Next application is non-invasive blood glucose monitoring. Near-infrared spectroscopy is used across the ear knob to measure glucose. Amount of near-infrared light passing through the ear knob depends on the amount of blood glucose in that region. The ear knob was chosen due to absence of bone tissue and also because of its relatively small thickness. Other applications include determination of unknown contaminants in industry using FTIR, determination of cell wall of mutants and wild type plant varieties using FTIR, biomedical studies of human hair to identify disease state, identify odor and taste components of food, determine atmospheric pollutants from atmosphere itself, and last is the examination of old paintings. It is also used in the forensic analysis in both criminal and civil cases. Example in identifying polar degradation, in determining the blood alcohol content, etc. Chemical analysis testing pill quality according to Medical News Today. Scientists at the University of Maryland have been successful in using the method of near infrared spectroscopy to make a prediction regarding quick dissolution of pill inside the body. Strength and limitations. IR alone cannot determine a structure. Some signals may be ambiguous. The functional group is usually indicated. The absence of signal is definite proof that the functional group is absent. Correspondence with the known samples IR spectrum confirms the identity of compound. Thank you.